Hi all, in this video we are going to see the history of crop genome sequencing, and here I would like to give the brief outline on genome sequencing and the techniques involved, the crop sofa sequence, and also we got to see the future perspectives of genome sequencing. And what is this genome sequencing? Genome sequencing refers to the method of determining the order of adenine, guanine, cytosine and thymine in the genome of an organism. There are several procedures for DNA sequencing, the most popular being the chain termination method, which is being first devised by Fritz Sanger and his colleagues in mid-1970s. Sanger's method of DNA sequencing is also known as chain termination method. In this method, dideoxynucleotide triphosphate is used as the chain terminator. This method requires single-stranded DNA, DNA primer, DNA polymerase, radioactively or fluorescently labeled nucleotides, and modified nucleotides that terminate DNA strand elongation. Here, the DNA sample is divided into 4 separate fragments and the sequencing reactions were followed. One of the 4 dideoxynucleotide, which are the terminators and terminate the strand extension and resulting in DNA fragments of varying lengths. Then the labeled DNA fragments are heated, denatured and separated by size by the help of gel electrophoresis and visualized by autoradiography or UV light. An improved version of this chain termination method is the di-terminator sequencing method. Now we're going to see the another method which is developed during the same period by Maxim and Gilbert. In 1976-77, Alan Maxim and Walter Gilbert developed a DNA sequencing method based on chemical degradation at specific location of the DNA molecule. The end-labeled DNA fragments are subjected to random cleavage at adenine, guanine, cytosine and thymine using specific chemical reaction. But by comparing the Sanger's method and Maxim and Gilbert method, Sanger's method is more efficient because it avoids the toxic chemical and it is also the ruling methodology in the whole genome sequencing. With the chain termination method, up to 96 sequences can be obtained simultaneously in a single run of a sequencing machine. This is still not enough to fully satisfy the demands of genome sequencing, and an alternative method called pyrosequencing has become popular. Pyrosequencing, which was invented in 1998, forms the basis of the massively parallel strategy that enables the hundreds of thousands of short DNA sequences to be generated at the same time. This method does not require electrophoresis or any other fragment separation procedure. Now let's see on the next generation sequencing techniques. The pyrosequencing, which was developed by life sciences, is also a next generation sequencing technique. Illumina sequencing, solid sequencing, DNA nanoball sequencing technique, helioscope, single nucleotide sequencing, and SMRT sequencing, RNAP sequencing are the next generation sequencing techniques. Now let's see in detail on Illumina sequencing. Illumina sequencing is the highly used NGS strategy for plant biotechnology and breeding. In Illumina sequencing, the first step followed is sample preparation, which involves extraction and purification. Next to sample preparation is the tagmentation, in which the adapters are linked to the DNA insert. Clustering is a process where each fragment is isothermally amplified and then hybridized, which is followed by bridge amplification. Then the reverse strands from there are washed away. The forward strands are only sequenced. The sequencing by synthesis is the strategy followed here for sequencing. After sequencing, the sequences from cluster library is aligned to the local sequence clustering forms contiguous sequence. Then these are again aligned to the reference sequence for variant identification. Now let's discuss on whole genome sequencing. There are 2 approaches for the whole genome sequencing, which are the shotgun approach and clone contig approach. Shotgun approach, the whole genome is randomly broken into short fragments. Then the resulting sequences are examined for their overlaps and these are used to build up the contiguous genome sequence. And then let's discuss on the clone contig approach. which involves a pre-sequencing phase during which a series of overlapping clones is identified. This contiguous series is called as contig. Each piece of cloned DNA is then sequenced and the sequence is placed at its appropriate position in the contig map in order to gradually build up the overlapping genome sequence. Now let's discuss on the history of the crop genome sequencing. Arabidopsis. Arabidopsis is the first plant to be sequenced in 2000. It's over 10,000 years since the genome sequence of the first crop was published. Since then, the number of crop genome sequences each year has increased steadily. The amazing pace at which genome sequences are becoming available is largely due to improvement in sequencing technologies both in terms of cost and speed. Sequencing the Arabidopsis model plant genome in 2000 was a major milestone not only for plant research but also for genome sequencing. It was among the earliest genomes from multicellular organisms to be completed and was sequenced by a large multinational consortium to cope up with this daunting effort. It was sequenced based on the back cloning. The back sequences were individually assembled and arranged according to physical map, creating a very high quality genome sequence. Effort and time associated with this approach limited its applicability to a few genomes. Next comes the rice. Only after 2 years later, the approximately 3 times larger genome of the first crop plant, rice, was elucidated using the same back cloning approach in 2002. And the genome size of the rice is 430 megabase pair. Next, the popular. Popular was the next crop genome sequenced and employed the emerging whole genome shotgun strategy. Here, the genome is randomly broken down into smaller pieces which are then sequenced and subsequently assembled. This strategy lowers the preparation cost and time. Now, grapevine. In 2007, grapevine genome sequence was published independently by two different groups, both based on the WGS strategy, which means the whole genome sequencing shotgun strategy. In 2008, the papaya has been completely sequenced. In 2009, sorghum, maize and cucumber have been sequenced. Sorghum was sequenced by Sanger method, cucumber by hybrid method and maize by following back-by-back method. In 2010, soybean and apple were sequenced. Soybean by following Sanger method and apple by using hybridization method, that is by Illumina technology. In 2011, cabbage, cocoa, wild strawberry, potato were sequenced. Cabbage by Sanger method, cocoa and potato by hybrid method and wild strawberry by NGS strategy. In 2012, barley, banana, tomato, muskmelon and watermelon were being sequenced. Barley was sequenced by hybrid method, banana and tomato is also being sequenced by hybrid method, muskmelon and watermelon by NGS method. In 2013, orange, chickpea and peach were sequenced. Chickpea and orange were sequenced by NGS method and peach by following Sanger method. Draft genome sequence of oilseed species castor bean Ricinus communis has been completed in 2010 across 10 chromosomes is estimated by flow cytometry to be 320 MB approximately. In 2012, the draft genome of Pigeon P. cajanus cajan was completed by Illumina GA and high sequencing 2000 sequencing methods. Based on KMS statistics, it is estimated to be approximately 833 MB. During the same year, the genome sequence of foxtail millet was completed by using whole genome shotgun next generation sequencing strategy. The genome size was estimated to be approximately 490 MB by cytogenic methods. Mung bean. Vigna radiata, variety radiata, was sequenced by using Illumina HiSeq 2000 in 2014, which insights into the evolution within Vigna species with high quality draft genome with genome size of approximately 579 MB. In 2015, sequencing of Gossypium hirsutum, a allotetraploid cotton, providing a resource for fiber improvement, was made by Illumina sequencing and more than 97.7% of genome was covered by more than tenfold parent sequence reads. Wheat genome sequencing. In India, the most important staple crop next to rice is wheat. International Wheat Genome Sequencing Consortium was created in 2005 by a people of scientists, breeders, public and private breeders. It has more than 1,500 members in 60 countries. Sequencing the wheat genome, once considered by scientists to be an insurmountable task, has been achieved through a worldwide collaboration of researchers spanning 13 years and been published in August 2016. Wheat genome consists of 21 chromosomes, which is more than 5 times larger than human genome and has identified position of 107,891 genes, including 4.7 million molecular markers, as well as sequence information between genes and markers, which contain elements influencing gene expression. Pearl millet, Centris americanus, was sequenced and published in October 2017 by using whole genome shotgun sequencing strategy and bacterial artificial chromosome sequencing. The genome size was found to be approximately 1.79 GB with 38,579 genes. Capsicum annum. Linked read sequencing technology has recently been employed successfully for de novo assembly of human genomes. Using this technology, 3.5 GB genome of Capsicum annum was sequenced in 2018. Through this video, we have covered the genome sequencing history of major economically important crop species. Hope you all enjoyed this video. Thanks for watching.